Hello, I'm Professor Sims, and in this video I will discuss microbial genetics, DNA extraction, and in vivo replication. This is the 9th in the series of 10 lab sessions held as part of my laboratory for the Fundamentals of Microbiology course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include learning about the structure and function of DNA, understanding the process of in vivo DNA replication, performing a DNA extraction using chemical methods, and of course understanding safety and disposal procedures relating to this lab. In 1952, Rosalind Franklin used X-ray diffraction to understand the structure of DNA. It was Franklin's scientific expertise that resulted in the production of well-defined images that would clearly show the double helix structure of DNA. Watson and Crick described the DNA molecule from Franklin's X-ray. Watson and Crick proposed that DNA is made up of two strands that are twisted around each other to form a right-handed helix. The two DNA strands are antiparallel, such that the 3' end of one strand faces the 5' end of the other. Austrian biochemist Erwin Chargaff examined the content of DNA in different species and found that the amount of adenine was very close to equaling the amount of thymine, and the amount of cytosine was very close to equaling the amount of guanine, or, in other words, adenine binds with thymine and guanine binds with cytosine. The X-ray diffraction pattern of DNA, also known as the photo 51, shows its helical nature as photographed by Rosalind Franklin in 1952. Analysis of the diffraction patterns of DNA has determined that there are approximately 10 bases per turn in DNA. The asymmetrical spacing of the sugar phosphate backbones generates major grooves where the backbone is far apart and minor grooves where the backbone is close together. So this here is an actual photograph taken by Rosalind Franklin in 1952 for the first time ever, showing the structure of DNA. So DNA, which is also known as deoxyribonucleic acid, is in fact made up of nucleic acids. The building blocks of nucleic acids are nucleotides. A nucleotide contains a pentose sugar, a phosphate group, and a nitrogenous base. Deoxyribonucleotides within DNA contain deoxyribose as the pentose sugar. There are pyrimidines, cytosine and thymine, or you can have purines, which are the adenines and guanines. These nucleotides are linked together by phosphodiester bonds between the 5' phosphate group of one nucleotide and the 3' hydroxyl group of another nucleotide. A nucleic acid strand has a free phosphate group at the 5' end and a free hydroxyl group at the 3' end. So this is what it looks like. The nitrogenous bases adenine and guanine are the purines, and they have a double ring structure with a 6-carbon ring fused to a 5-carbon ring. The pyrimidines, cytosine and thymine, are smaller nitrogenous bases that have only a 6-carbon ring structure. So the pyrimidines have a longer name, but a smaller structure, and the purines have a smaller name, a larger structure. The 3 components of a deoxyribonucleotide, are the 5-carbon sugar here, called deoxyribose. They have a phosphate group and a nitrogenous base, a nitrogen containing ring structure that is responsible for complementary base pairing. So here, the base is going to be one of these guys. Either cytosine, thymine, adenine, or guanine is going to act as the nitrogenous base. And it binds to the deoxyribose sugar, and it is bound to a phosphate group. So the whole thing here is the deoxyribonucleotide. And then the 3', 5', and all that stuff has to do with this structure here. So this is the 5' carbon on this sugar. So this here is the 5' phosphate group, and then this is the 3' carbon of this sugar. So this is the 3' hydroxyl group. So now you have phosphodiester bonding between the different nucleotides, and this is what forms the sugar phosphate backbone. The alternating sugar phosphate structure composing the framework of a nucleic acid strand. These covalent bonds that are occurring along the outside of a DNA molecule are fairly strong, whereas the hydrogen bonds that are formed on the inside of the DNA molecule are relatively weak. The interior of the DNA has adenine and thymine that forms hydrogen bonds with each other, guanine and cytosine forming hydrogen bonds with each other. Adenine and thymine have 2 hydrogen bonds holding them together, whereas guanine and cytosine have 3. This is a triple bond and double bond. So the overall structure of DNA kind of looks like a twisted ladder. That is, the sugar phosphate backbones are on the outside of this double helix, and the purines and pyrimidines on the inside kind of form the rungs of a ladder. Sugar phosphate backbones and adenine, thymine, cytosine, guanine on the inside. These are phosphodiester bonds along the outside that are pretty strong bonds, and then relatively weak bonds in the middle, these hydrogen bonds. The two DNA strands are anti-parallel to each other. This is going 3' to 5'. This is 3' to 5', and remember those numbers come from the deoxyribose sugar and where things are bound, which carbon they're bound to. So it's really important to understand the DNA structure before you try to understand how DNA is replicated. And replication is very important. Without it, there would not be any life on Earth. Your cells have to divide. And before cells can divide in vivo meaning in nature, they have to double their cell structures. They have to double all of the organelles and their genetic information. And that is done, the genetic information is doubled using DNA replication. So first, the double helix of the DNA separates or unzips along those weak hydrogen bonds in the middle. Those bonds between the adenine, thymine, cytosine, and guanine are pretty weak, and they can be split apart. Then you have free nucleotides, which are present in the nucleus, that will attach themselves to those now exposed nitrogenous bases. They can attach by hydrogen bonds to the exposed bases, and then two identical strands will form from each single strand of DNA. And this here is a good simulation on DNA replication. You do want to make sure that you become familiar with all of these enzymes and their roles in vivo DNA replication. This will become especially important when we talk about in vivo replication versus in vitro replication in Lab 10. So in vivo, in nature, helicase is the enzyme that breaks apart the hydrogen bonds in the middle of the DNA strand. Topoisomerase is the enzyme that helps to protect the sugar phosphate backbone while all of this is happening. Primase will create RNA primers. DNA polymerase catalyzes the addition of new nucleotide bases. And then DNA ligase will join together newly synthesized fragments. So when studying DNA or performing molecular genetic research, the first step is usually going to be extracting and purifying the DNA to be investigated or manipulated. There are several methods commonly used to extract DNA from specimen. Some physical methods include beads or rotor mills, liquid homogenation, manual grinding, sonication, or freeze-thaw cell lysis, which is what we use in my on-site labs. Some chemical methods include detergents, enzymes, and chiefly what we're going to be using is osmotic shock. For experiment 1, you will use osmotic shock and detergent to extract your DNA. You're going to be extracting it from your epithelial cheek cells. Osmotic shock occurs due to the pressure on the cell membrane that comes from differences in salt concentration on opposing sides of the membrane. So you can have a hypertonic, hypotonic, or an isotonic solution. Isotonic is when everything is in equilibrium. The salt concentration is the same on both sides of the membrane, everything's happy. But in a hypertonic solution, you have concentration outside of the cell that exceeds that inside of the cell. So water is diffusing out of the cell via osmosis, right? So all the water is leaving the cell, to try to achieve equilibrium. In a hypotonic solution, the flow of water is reversed, causing the cell to swell and potentially lyse. Here is a short video illustrating these concepts.
We're going to explore what happens when you place a cell in a solution that is much more salty, hypertonic, much more watery, hypotonic, or has an equal salt concentration, isotonic. Remember that cell membranes are selectively permeable. They allow certain particles to pass through, but not others. Usually, larger molecules can't fit through the membrane without special channels. The cell membrane is fitted with special protein channels called aquaporins, which allow water molecules to pass through without expending energy. However, the ease with which the water molecules can cross is going to depend on something called the concentration. Concentration is a measure of how much solute there is per volume of solvent. In other words, whether the liquid is more salty or more watery. We'd usually expect that the molecules would follow a process called diffusion. They would flow from areas of higher to lower concentration. Eventually, they will reach equilibrium, where there will be an equal concentration on both sides of the membrane. However, in the case of a liquid like salt water, the salt molecules are much too big to fit through the membrane, so the only particle that can move is water. Water molecules that are not stuck to a molecule of salt are far more likely to relocate. In this first scenario, the liquid surrounding the cell has many molecules of solute in it. This liquid is hypertonic compared to the cell. It has a much higher concentration of solute particles and a much lower concentration of water. Unfortunately, the solute molecules can't pass through the membrane to reach equilibrium, but water molecules can. The molecules on the inside have much less solvent getting in the way. Water will begin rushing out of the cell. Some water will come in, but the net movement, the overall movement, of the water molecules will be outwards and the cell will shrink. Now let's immerse a cell in a much less concentrated solution, a hypotonic solution. Compared to the cell, it's much less salty. Some water molecules will exit the cell, but even more will rush in because they aren't obstructed by solute particles. The cell grows in size, and it may even undergo cytolysis and burst. Placing a cell in an isotonic solution, where the concentrations inside and outside are equal, is much more pleasant. Equal amounts of water molecules continue to pass in and out of the cell. The system is in dynamic equilibrium. Particles are continuing to move, but the net movement is zero. The cell neither grows nor shrinks and is much more likely to survive.
One of the first steps in experiment 1 is that you're going to be gargling salt water for 1 to 2 minutes. And I know that's a long time. But you do this to release the cheek cells from the endothelial membrane of your mouth. And you're going to want to get a lot of them because you're going to want to be able to observe the DNA at the end. So you gargle the salt water for 1 to 2 minutes. And also having all of this salt in your mouth is creating a hypertonic solution, which is helping in the process of osmotic shock. The dishwashing soap, obviously, is a detergent, and detergents are generally used to denature proteins. Denature just means to break down. So this is helping to purify the DNA that you're extracting. You will be able to observe your DNA in the isopropyl alcohol, and they're going to look like white filamentous strands that are floating around on top like this here. So you have detailed steps located in the Lab 9 protocol for performing this extraction, but here is a video showing the procedure in a nutshell. The next step after extracting the DNA is usually going to be to perform some quality control. That is, you need to make sure that you first have the DNA, you have enough of it to use for further experiments, and that the fragment that you wanted to get, for example, if you're looking at a specific set of genes or a specific genetic marker, you want to make sure that it's the one that you wanted, and you wanted to make sure that it's pure as well. It's not got a bunch of proteins and lipids and stuff mixed in there. And this is done using gel electrophoresis. Gel electrophoresis is a technique commonly used to separate biological molecules based on their size and charge. Due to its negatively charged sugar phosphate backbone, DNA is strongly attracted to a positive charge. So, an agarose gel is oriented horizontally in a buffer solution such that the DNA samples are loaded into the sample wells here on the side of the gel closest to a negative electrode, and then voltage is put across it, and the DNA is drawn through the molecular sieve of the agarose matrix toward a positive electrode on the other side of the gel. The agarose matrix impedes the movement of larger molecules through the gel, whereas smaller molecules pass through more readily. So the distance of migration is inversely correlated to the size of the DNA fragments. That is, the smaller fragments travel a longer distance than the larger fragments. Sizes of DNA fragments within a sample can be estimated by comparing them to fragments of known sizes in what is called a DNA ladder or a marker ladder. What you do is you run your DNA of unknown size alongside a marker ladder, and this is going to be DNA that has been cut at very specific sites with restriction enzymes, and they're cut into fragments of known length. And when you run the marker ladder or the DNA ladder, you're going to know what size these bands are. The bands that are at the top are usually larger, and the ones at the bottom are smaller. So you might have this is 1,000 base pairs, and then it goes 800, 600, on and on and on until you have like 50 base pairs, something like that. You can compare the bands that you get from your experimental DNA to the bands that are in the marker ladder to see about how big or small they are. For experiment 2, you will complete a simulated electrophoresis gel. You want to make sure you record the procedural steps and be able to interpret a gel image. Note that the gel not only tells you the size of the DNA by comparing to the marker ladder, it also tells you if you have DNA at all, for one thing, if you have a band at all. And then it also can tell you how pure it is, how much of it, how concentrated it is, based on how crisp the bands are. If you have nice, crisp, sharp bands like these, then you know that that DNA is pure. If you had a bunch of impurities in the DNA, then you would have like a big, thick smear going all the way down the gel. And you wouldn't even really be able to see where the band is because it'd be so smeary. Thick, bright bands, like these guys here, are real thick and bright. That means that that DNA is highly concentrated. There's a lot of it. So there's a lot more DNA in this band than there is in this one. So for your observations for Lab 9, you want to make sure that you can explain Chargaff's rules, the A to T, C to G stuff, how nucleic acids are bound together in DNA, how strong the bonds holding together deoxynucleotides versus the bonds holding together the sugar phosphate backbone. So you need to understand the difference between the phosphodiester bonds and the hydrogen bonds, which enzymes pull the DNA strands apart, which enzyme makes the RNA primers, which enzyme adds new nucleotides to the parent strand, and which enzyme joins together Okazaki fragments. You need to know all of those. And also make sure that you know that this is in fact in vivo, the in vivo DNA replication. And it's going to be important to know that and have that distinction because next lab you're going to be talking about in vitro, which is artificial replication. Were you able to successfully extract and view your DNA? What do you know about osmosis and detergents and how these were used to make this procedure possible? And then what information does performing a gel electrophoresis yield? Explain what the gels tell you and how you can observe those results. For experiment 1, you're going to use the same safety precautions you would for the other wet lab procedures. Even though you're not using anything especially harmful, remember that you should treat every organism as a potential pathogen, and don't forget to follow the safety guidelines and disposal procedures previously outlined for the microbial stains and glass slides as well. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics and leave your questions for me in the comments below.